Hey folks, in this video we're going to start talking about the creation of energy. And maybe it's not surprising to you that energy is the ability to do work, because when you don't have energy, you're not really able to do much work, are you? Your body primarily uses energy in terms of ATP. And the way I think of ATP, I think of it as kind of a mom going to the grocery store and trying to wrangle 3 children at the same time. This is how I grew up, and it was really difficult for my poor mom. When she was able to leave one of us at home, now she actually had more energy to herself because she wasn't expending so much energy holding all of them. This is what happens to the ATP molecule. The molecule expends a lot of energy trying to hold on to its 3 phosphates. And in trying to hold on those 3 phosphates, it's using so much energy that when we break one of them off, there's a release of energy. That energy can then be used for other functions. One of those we saw in our previous unit when we talked about the movement of ions across a membrane and how sometimes they can be pumped by channels that are using ATP. When we talk about potential energy, that's like the potential for something to do work. What this means is if you had a ball that was sitting on top of a hill, that ball, if you pushed it, would roll down the hill. The potential energy is basically that the ball is at the top of the hill and it's able to roll down. It's just not rolling yet. When it is rolling, you could capture that energy and use it for something. We saw that a little bit in the previous unit where you were using a molecule going from its high concentration gradient to its low concentration gradient to move another molecule from low to high. The one that was going high to low had potential energy before it started moving. Once it started moving, now it was releasing energy, and that was able to be captured by another molecule that wanted to go from low to high. When your cells are making energy, they use glucose. And basically what they're doing with that glucose is they're kind of splitting it apart into ever smaller molecules. And as they do that, energy is going to be released from the bonds. They use oxygen in this reaction to create even more ATP. The byproduct of this reaction is that some of those carbons from the glucose and some oxygen comes off. So basically the cell is using glucose and oxygen, and it's creating ATP and a waste product, carbon dioxide. I'm going to warn you that this is one of the more complicated things that we study as biologists. And for a lot of people, it would have them looking like this. But you're not going to be like that, because we are going to work it out together. So cell respiration can really be broken into 4 main components. The first one is called glycolysis, and that is present in a lot of different kinds of cells. The second one is called pyruvate processing, and the name will make a lot more sense once you understand the process. The third one is known as the Krebs cycle, the citric acid cycle, or the TCA cycle, and it just depends on what source you're reading from. And the fourth one is called oxidative phosphorylation. We're going to start with glycolysis. And in glycolysis, the big thing that you need to learn is that we're going from glucose, which is a 6-carbon ring sugar, and we're breaking it down into 2 3-carbon sugars. So we start with a 6-carbon sugar that's a ring. The first thing that we do is we use an ATP, and that ATP is going to break that 6-carbon ring it's going to turn it into something linear with still having 6 carbons. The next step is that we take an isomer of it. We just make it look a little different. After that, we add a phosphate. The addition of that phosphate takes ATP. Then we add another phosphate. The second phosphate makes it really unstable. Once it's unstable, guess what happens? It can break into 2 3-carbon sugars. So right now, this is going to seem like a really raw deal. Basically, in trying to make ATP, we have spent 2 ATP, made none, and all we've accomplished is we've broken a 6-carbon sugar into 2 smaller 3-carbon sugars. But now is where the fun happens. Basically, what we're going to be doing in these next couple steps of glycolysis is generating energy. In the first step, what we're going to do is we're going to oxidize one of those 3 carbon sugars, and that reduces something called NAD, which moves it into NADH, which is an energetic molecule because it's holding onto those electrons. It can do something with those electrons. The way that we do that, that oxidation is that we add a phosphate. So if you're keeping count on our 3 carbon sugar, we have 1 phosphate from making it unstable, and now we have another phosphate that we added during that reduction. Well, guess what? Those phosphates can go somewhere. The full name of ATP is adenosine triphosphate. So hopefully now you're starting to put it together that what we can do is we can add one of those phosphates to an adenosine diphosphate to make an adenosine triphosphate, our energetic molecule. So sure enough, what do? We take that 3 carbon sugar that's got those 2 phosphates on it, we pop a phosphate off, make an ATP, and in another step, we pop another phosphate off and we make another ATP. At the end, we have a sugar called pyruvate, which is a 3 carbon sugar. So let's kind of look at the energetics here. Remember in that first step where we were breaking apart our glucose into 2 3 carbon sugars, we spent 2 ATP. As we were reducing one of our 3 carbon sugars, we made a NADH, that's an energetic molecule, and 2 ATP. So basically where we're at now is we spent 2 ATP, but we made 4 ATP and 2 NADs. Now, where am I getting those numbers from? Well, we split it into 2 3 carbon sugars, so we do that cycle twice. Whew. So if that last one seemed really complicated, this next one's not going to seem that bad to you. This step is called pyruvate processing, and during pyruvate processing, what we do is we take our 3 carbon sugar, chop a carboxyl group off of it, oxidize it. In oxidizing it, we end up releasing a NADH, the energetic molecule of NAD, and then we also release some CO2. Now remember, we're doing this with both pyruvates, so we do it twice. So we will make 2 NAD. All right, it's NADH, but I'm still having fun with it. Then what we do is we add an acetyl to the party, and that turns our pyruvate into something called acetyl-CoA, because now it's been added onto that molecule. When we move into the citric acid cycle, the first molecule you're going to start with is that acetyl-CoA. So if you're wondering where it goes, that's where it goes. Now, it's important to remember when we go through the citric acid cycle that you get 2 turns for every glucose, because remember, we broke that glucose into 2, and that means we made 2 pyruvates, which led to 2 acetyl-CoAs. And that means we're going to be able to go through that process of the citric acid cycle twice.